All right. So I haven't got a chance to grade your exam yesterday. I'm disappointed because I actually wanted to get to at least the scientific method assignment this morning and get that graded before class, but I had a barrage of emails. You know how that works. However, I will be working on that, and as I said in class, the latest you can expect to see both of those graded would be, by the end of the day on next Tuesday. And I know that means you got to wait the weekend, but trust me, I'm not one of those people that likes these things sitting around. I know that you guys are anxious about getting your grades back, and especially in a mini semester. So I will work diligently. I will get on them today. I don't know how far I'll get through them, but I will definitely get those done by, I said, next Tuesday. For those of you who student athletes need to have the evaluation done, I don't know what the due date is. I'm going to email coach and find out. But if you know what the due date is, please let me know, because I want to kind of wait till after I grade your exam before I do those. So, all right, that's all as far as announcements and all that kind of stuff goes. You guys have anything? You two have anything that you want to talk about? Let me get the lecture loaded up while I'm thinking about this. Anything that we need to discuss? Any problems, concerns? Where were we? That picture I was showing you when we first got online, I'm just thinking now the recording I started after the picture, but I actually put it in the chat box. Let me get myself arranged here. Where did we go? Where did we stop the other day? There? No, we didn't do that. Oh, we didn't even, we went, there we go. Here, right? Did we stop here, the mitochondria? I'm trying to remember with the cell, or was it this one? It might have been this one. I hear your doggy in the background.
Oh,
sorry. Where did I move? Okay. No. Why didn't I make myself a note? I usually do. See, there you go. Okay. I think this is where we left off. No. Endomembrane system, right? There we go. That's where we were. Yes, the last thing I remember saying to you guys was we went back and we looked at the nucleus, and I showed you, and I did get my pen to work finally because the battery had gone dead. Quadruple A batteries, you can't find them anywhere. I had to buy it on Amazon. But you'll remember I circled this little area right here, and I wanted to remind you that, as the name tells you, hi, Tim. How are you this morning? How's your eye? That it is an endomembrane system. So it's a system of membranes. Remember what membranes are made up of, right? It's those phospholipids in a bilayer. I'm trying to kind of hammer that unity and diversity thing. Good, good. I'm trying to get you guys to remember that there are connections, and that's important, form and function, right? So these membranes, the one in the blue, that's the rough ER, and then the one in the purple, which is part of that nuclear envelope. Remember, that's like a double membrane, right? It's got an inner and an outer. All right, so all of those things are connected together. They're all made of the same thing, and they're all connected together, right? And that's really all I want you to get out of this picture, because we're going to run with this. We're going to go with this, but I want you to kind of make that that visual and that realization that the reason why we call this endomembrane system is because it's a transport or a cycling of membranes. That's a better word. Cycling. Don't use transport. It is a transport, but cycling means you're going back and forth, right? Okay, so you understand the purpose of the endomembrane system. Let me go back to the previous slide so we can kind of get this idea, like like you do at the mall, where you go, you are here. Except, where am I? I'm lost. I'm over here by the shoes. No shoes. There we go. All right, so we just talked about the nuclear envelope. Let's now talk about that great thing with that incredible name, right? The endoplasmic reticulum. I love that. I love to say, oh my gosh, the other day I got up and, when I woke up first thing in the middle of the night and the light was off and I had to go use the restroom, and I reticulated and ran into the side of the bed, put a big bruise on my shin or something like that. People go, you're so weird. All right, let's take a look at the nuclear envelope again. See what I just said? It provides a pore, an opening. What does it provide a pore for? Oh, I see what it provides provides a pore for? It provides a pore for something to get out that might be involved in delivering a message, right? Sure, like the mail. In this particular case, and we'll talk a lot more about this molecule later on, folks, so don't worry about going, what's it doing again? All right, messenger RNA. That's what that pore is for, that little thing that I circled. There it is, little smaller picture, but there it is. And remember, there are lots of these all over. There's not just one, right? So the messenger RNA leaves through that opening in the nuclear envelope, which is really not an opening so much as a pipe, a conduit, right? Because it turns right into the next type of membrane. How? Well, it's the same stuff. It's all phospholipids in a bilayer, right? This is really more just a matter of location than anything. You're either part of the nuclear envelope or you're part of this this endoplasmic reticulum envelope, okay? All right, now if you want to go through and kind of figure out what it is they're referring to here, the lumen is the opening. So if you think about this as being pipes, right? And if you see the way it's drawn up here, again, I'll get to the smooth and the rough in a second, but that's what they mean by the lumen. And I'm trying to figure out what they're calling the cistern there, but it looks to me like the cistern is the space between the lumen, right? So it looks like the lumen is that space they're pointing to here. That's it, Steve, color blue on blue, right? And then it looks to me like the cistern. Lumen is a word that you'll see. Cistern's a word you'll see a lot, but lumen is something that I think you'll see more often. So lumen is an opening. A pipe has a lumen, okay, has an opening. All right, endoplasmic reticulum. What is this all about? All right, well, it's membranes, and as I said, there's two types. For no really fancy reason other than the fact that under a microscope, they look like the name says. They're either rough, as I point to the smooth, excuse me, they're either smooth or they're rough under a microscope. See all the squigglies on the left and the right? Those are all endoplasmic reticulum. You're doggone right there. They're all endoplasmically reticulating, aren't they? Now, the difference between the rough and the ER, other than the fact of the way they look, the reason why the rough ER is rough is because there's something attached to them, right? That's what makes them look. You can see them. You can see them in the picture, the little tiny dots, little tiny black dots. Those little tiny black dots, those are ribosomes. That's why the rough ER looks rough under a microscope, because it's got ribosomes attached to it. You remember talking about ribosomes? We talked about ribosomes with with prokaryotes, with the bacteria, didn't we? What do ribosomes do? What's their job? Man, it's just D3 this morning. I must have really scared everybody away with the exam. Holy cow. I haven't even finished grading it. Or for that matter, even started. Yeah. And Tim, what do you need to synthesize a protein properly? What is the ribosome going to need and make sure it does that? Okay, that's right. But DNA is a really, really special molecule, right? So the DNA, which is think about, is the archive of the code. The DNA really doesn't do any work, right? Other than hold on to the code. It's such a special, important molecule that it really never leaves the nucleus. It stays protected inside of there. So I'm kind of thinking more about the intermediary between those two things you mentioned. Trying to think of, who would be responsible for delivering that message. Yeah, right? Messenger RNA. Now, winner, chicken dinner, we go all the way back. Look at that nuclear pore. Notice how that happens? The messenger RNA, which again, we'll talk about how that comes about later on, goes out through the pore, finds itself a ribosome, and ding, 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 ding. That's the factory, right? Ribosome starts making proteins, right? The bound ribosomes specifically, I moved my attention up to here as it bounces around, of course. Why is that pen looking weird? Anyway, the kind of bound ribosomes produce things called glycoproteins. Glyco. I know I've seen that root before. Where have I seen glyco before? What does glyco mean? Glycogen is where it's at. Here in the next couple of chapters, we're actually going to see a word like this. Oh, to split glyco? Let me know what glyco is. Glycocardiac on February 14th. Right? February 14th, you can give a card to your glycocardiac. Oh my gosh, Steve, that is pathetic. I like that, though. What's a glycocardiac? Cardiac. It's not an A&P class. Come on, guys. Yep. And glyco's sugar, sweet. So on February 14th, you'll do something for your glycocardiac, won't you? You do something for your sweet heart. Okay. Rim shot, right? That's what kind of proteins are being synthesized in the rough ER. Glycoproteins. Okay, a glycoprotein is a sugar and a protein. Nobody says you can't stick those molecules together. I mean, yes, we talked about monomers making polymers in each group, but come on, folks. You can also make these massively big molecules that have sugars and proteins stuck together. Anything that's sticky, for whatever reason, will stick together. All right, what else does the rough ER do? The rough ER produces another part of the endomembrane system. I don't want to keep bouncing back to that original slide with all the names, but there was one on there. Good morning, Wyatt. There's one on there called the vacuoles, okay? Now, if you remember, the vacuoles were empty sacs. The water vacuole holds water. I said there were pigment vacuoles. Okay, there's another word for vacuoles, but it generally refers to maybe a smaller vacuole, and that's something called a vesicle. Excuse me. I wanted to emphasize, though, that, as I said, it's another word for a vacuole. What I mean by that is vesicles, vacuoles, these are things surrounded by a membrane. Same membrane we've been talking about all along. Anytime I say membrane, you go, oh, phospholipids in a bilayer. But these vesicles are smaller because many times what vesicles do is they transport things, okay? So the central water vacuole in a plant cell doesn't transport anything. It stores water. But these things act like the maybe the post-delivery folks of the cellular world. You notice the little red arrow in the picture? Look how this is happening. Isn't this cool? What we're doing, Cooper, you mind? You don't need to make your bed right now. So funny how dogs with that instinctual, just like wolves, will scratch. You know, they do that in the wild to make their bed, and you're like, but it's a bed. Everything's nice and comfy. It's a big comforter, Cooper. Relax. We're all driven by instinct, aren't we? All right, so I was back to what I was saying. How do we get from what's inside the ER? And if you're thinking, what does it mean, what's inside the ER? Well, remember what's attached to the rough ER. What's attached to the rough ER are ribosomes. So what's inside the lumen? I think I introduced you to that word, right? Proteins or glycoproteins. That's why we say the rough ER secretes them. And how does it secrete them? Ba-ba, with transport vesicles. And how do you form a transport vesicle? You pinch it off. Look in the transitional ER. Notice what's happening. As the proteins gather at the end of the ER, the rough ER, they begin to form a bubble. And that bubble grows, and that bubble grows, and that bubble grows until they could have drawn from either one. It pinches off. Just like you take a wad of dough or clay and squeeze it, right? Bloop, plop, comes off, or toothpaste, or whatever analogy you want to use. It makes sense, right? And as that membrane pinches off, it pinches off the material inside. So inside that transport vesicle, there are glycoproteins. Okay? Now, we're not done with their journey yet. In other words, we're not finished with the endomembrane system, but I do want to go back and talk about the other type of ER before we return back to what happens next week to the transport vesicle. Well, those are actually kind of cool. There's some neat stuff that they do. But anyway, smooth ER. There's another type of ER which is not responsible for protein synthesis, okay? Now, it does a variety of things. You can see I got it there. That's the smooth ER, right? Smooth ER is responsible for synthesizing fats, lipids, energy that you brought in, and is it in excess? Remember I said, think about lipids as being like your savings account, or maybe a treasury bond or something like that, right? Where it's for long-term energy storage. Now, we talked it has other reasons, blubber, to decrease buoyancy or increase buoyancy so you float, right? But all of that is going to occur in the smooth ER. Metabolizing carbohydrates, okay? So taking the sugars that you eat, the food that you eat, the carbohydrates that you eat, remember that's one of the classes of organic molecules. Those are going to be metabolized. They're going to be broken down. their energy is going to be extracted. That's what metabolism is, folks. I always say, physical is your physical. Metabolism is your, and I'm really kind of broadening this. And if you go on the A&P, you'll see what I mean by that. But that's your chemistry. Your metabolism is your chemistry. How this, as I've said before, and get off on this tangent, how this wonderful chemistry becomes living things, okay? So anyway, you got to have energy to do that. It's the Smoothie R. It's going to do that. A couple of chapters from now, we'll get into the chemistry of how that happens. Drugs and poisons, whether intentionally or unintentionally, we're running into this stuff every day. Some of the poisons that are produced in our, some of the poisons in our body are produced in our body, believe it or not. I'm sure you're aware of that. I'm not just talking necessarily about waste products. I'll talk about an organelle here in a couple of minutes that does all that. Calcium ions. This is where you store calcium ions. Anybody have any idea why calcium ions are important? Yeah, part of it is that. Anything else? That's a big part of it. Sure, absolutely. Oh, we cannot have our exam on Monday. Why did I think that? Holy cow. I'll take care of that. Let me write myself a note. Muscle contraction, nervous impulses, folks. Calcium ions are important. You go on in, again, if you do go on to A&P or other physiology courses, sorry, exam 2. I need to reschedule. Hold on. I will put an announcement. It looks like we're way behind, but the next 2 chapters go pretty quickly, actually. Yeah, that's right. Sure, which has to do with your electrical impulses, right? You get your calcium ions all messed up, your heart starts going, what is it? Arterial fibrillation. Instead of contracting, it just kind of shakes. No blood gets moved around that way. Okay, so Smoothie R. Any questions about that? And of course, there's that's a good compare and contrast kind of thing, the differences between them. But all right, we're back to our transport vesicle. Here we are. Wayne's, where the, you guys are too young for that. Well, some of you are. All right, going off into where we were with the transport vesicle. All right. Transport vesicles go a variety of places. Here's one place they go. They go to this thing and where they serve Italian food, right? When you want to go to a restaurant, Golgi's. Hey, you want to eat tonight? Yes, let's go on down to Golgi's. I love their pizza. That's definitely, I don't know where that word came from, but I'm guessing that's the guy who described it, and I'm guessing he was of Italian descent, because that's a really good, I wonder if there are any Italian folks named Golgi. So cool to meet somebody like that. Now, when I was in college, which was a long time ago back in the 70s, but when I was, no, it was the 80s. Anyway, when I was in the college, I remember my Bio 1 professor saying Golgi apparatus, Golgi complex, sometimes it's called. He said, we're not really sure what this does. He goes, at this particular point, let's just leave it open in your notes. That's what he told us. And I used to have my notes. I finally got rid of them, partially because they got rained on, partially because the cockroaches were eating them. But anyway, so in my notes it said that. All right, no, fantastic. This is what I love about science and biology. I get to teach new stuff. I get to teach all kinds of things that are new. Like, for example, the Golgi apparatus. Now we have a pretty good idea what it does. For one thing, those stuff that's made in the ER, here it comes. There's the transport vesicle. Now it's drawn as if it's cutting open. Inside are proteins, those little purple dots, right? So the Golgi apparatus is going to modify those. What's the most important thing about a protein? What's the most important thing about a protein? Yeah. So that folding and there are also things Mother Nature realizes the world is not perfect. So there are things that try to correct maybe proteins aren't folded exactly right. But anyway, all that sort of processing stuff is going to occur in the Golgi apparatus. So think about it as the maybe the shipping dock of the company where you work, right? Whatever you produce, if you're manufacturing, these are the guys that maybe package it for you and, send it off, right? Put the peanuts around it so it doesn't break. So there is part of the Golgi apparatus that is receiving transport vesicles, and that would be the cis face. And if you're saying, oh gosh, I remember that. Yes, remember geometric isomers? Geometric isomers, there was a cis and a trans version, wasn't there? Okay, and then the Golgi apparatus, by the way, is like the shipping dock. Those guys down there, those folks that work in the shipping, women, men, whatever the case may be, they don't want to keep the stuff you're making, the widgets, let's call them, right? They're there to ship them off. So that's what the Golgi apparatus does. Notice the big red arrow. Things move in on the sys face. They get modified, put in a packing slip. And on the other end, guess what happened? It pinches off and it forms another transport vesicle. So you see what I mean about that sort of ubiquitous word, transport vesicle? So that's what the Golgi apparatus does. It sorts and packages. Just like the mailing dock or the shipping dock in your company, the Golgi apparatus is also responsible for deciding what transport vesicles go where. And of course, if you think, how does it know that? It's all about chemistry again, folks, right? And again, story's not done because we want to figure out what happens to this. We've only made our way partially through the endomembrane system. We're near the end. So if you're thinking, oh gosh, how long does this go on? It goes on for a while, so it's kind of cool. But look at the picture of the Golgi apparatus on the right in the electron microscope. The Golgi apparatus is you can get online. I think I put one on Blackboard. To me, it's like if you were to drop oil into water, sort of those droplets that it makes. So the Golgi apparatus, the way it transports things through it, the way it transports those proteins through it is really not like in the ER. It's really not like where it goes through the channels. What happens is these layers form up here at the top and merge and then deform or unform, not deform, at the bottom, you see? So like if you're dropping oil this way, and then the oil droplets would kind of merge together, and then as they went on, they would kind of separate into smaller drops. So if you can visualize what I'm talking about, dropping some oil at the top of the water and kind of watching how it, remember I said oil and water don't mix, the bubbles just get smaller. Take a look at that video that I have on Blackboard, and if I don't, email me because I'm pretty sure I put one on there, but sometimes those links go dead. I'll see if I can find another one. Okay, so where are we, right? We're at the trans face. So now we're at the shipping site. So now we got a transport vesicle, and that transport vesicle is slated to go somewhere. And how is it slated? Well, as I said, it's chemistry, but depends on what's inside too, right? Transport vesicles, like the boxes at a shipping dock, can hold anything. So it depends on what's inside, what's going to happen to that transport vesicle. So I tell you what, why don't we look at a couple of examples of what that transport vesicle might be made for, right? Everybody follow me on this? Still on the endomembrane system. Okay, here's two places that transport vesicle can go. That transport vesicle can form something called a lysosome. Now notice, if this is confusing to you, I hope it's not, that just reinforces that some transport vesicles are lysosomes, okay? Vesicles, vacuoles, general term, it's a sac. Notice what I said in the next one. A lysosome is a membranous sac. That's just another word for a vesicle, vacuole, membrane. This word you ought to be familiar with. Hydrolytic enzymes. Maybe not enzymes, but things that engage in hydrolysis. So what is happening here? Okay, what is happening in a lysosome if there are enzymes that are engaging in hydrolysis? Are we building molecules or are we tearing them apart? Think about what the word hydrolysis means. Hydrolysis. Yeah, Beth and or Lindsay, how did you did you remember the word, or did you remember the water and the lysis part? Or both of them? Maybe you did. Hopefully both of them, because those roots again show up a lot, don't they? We've already seen lysis a couple of times, haven't we? All right, so take a look down below at a couple of the things that a transport vesicle full of hydrolytic enzymes might do. Well, one of the things they might do is break big chunks into little chunks. That's what Beth and or Lindsay just said. They tear things apart. So what a lysosome might do, full of these digestive enzymes, it might merge with, there's another one, a food vacuole. Guess what's inside of a food vacuole, right? And those two vesicles and or vacuoles will merge. All right, now stop for just a moment. What guarantees that the yellow-colored food vacuole's membrane will merge with the lysosomes, in this example, purple-colored membrane? Nah, too quick, Tim. What guarantees those things will merge together? I didn't say necessarily contact each other or identify each other, but I'm saying what guarantees that those two membranes of those two things are going to together like sticking two piles of dough together. You remember way back in chapter 2, I say way back, it was a couple of weeks ago, wasn't it? Didn't I say this? But not only more than the attraction. Tim is kind of talking about attraction and shape. That's why I'm trying to get off of that. But do you guys remember this little phrase I said? Hydrophilic dissolves hydrophilic, hydrophobic dissolves hydrophobic. They don't dissolve each other. We can kind of extend that phrase now to even talk about membranes too. Since they're all made of the same thing, the membranes of the lysosome, the ones in the purple, are made of the same thing that the membranes of the food vacuole are. So when things that are like touch each other, so beyond the attraction part, what do? Yeah, they become one. Oh, it was very metaphysical of you, very spiritual. They become one, right? When they merge. And guess what you've done now? When you merge those membranes, you turn those hydrolytic enzymes, those digestive enzymes, loose on that food particle. And that food particle is going to get chunked up into little tiny chunks, right? And that's all part of digestion. For goodness sakes, it starts in your mouth. When you chew, you break up little pieces into smaller pieces. Smaller and smaller and smaller and smaller and smaller till they become chemicals, which is the subject of the chapter in the future. If you're fascinated by this word, we'll talk about it later, but I'll give you a hint right now. Phago is to eat. Phago is to eat, and cytosis involves a cell. So basically cells eating there, right? Okay, back in high school, my buddy had an old 1972 Pinto, and one night, accidentally, the door was open, his driver's side door. When he backed up, I was in the car with him. I won't explain the circumstances, but nonetheless, he backed up his open door into a cement pole that held up a lamp at a local mall. It wasn't open. It was somewhere around 2 or 3 in the morning. But anyway, the door jamb got all messed up. We could close the door, but it wouldn't. So he and I eventually ended up going to a junkyard, and we bought a door and replaced it. All right, you follow my story? My question would be to you, we had nothing for money. We were both working at a scroungy little restaurant there in town. But would it have been better for him to buy another car monetarily, or just replace the door as we did? Which one would have made more sense financially, economically? Sure. Right? Sure. Now you're going, now I know where he goes with these stories. It's the same thing in a cell, right? Let's say an organelle has gone bad, like a mitochondria. You have several mitochondria in your cells. If you start working out, your cells will actually produce more mitochondria, or mitochondria will produce themselves in the cells, right? They will actually start doubling and making more of them because they're the things that take your food and convert it into energy. Now, we'll talk about what a peroxisome is in just a second, but a peroxisome is one of those things that it's interesting to me. That's what I was saying back with the ER. This is a poison that your body produces. Now, it has a way of getting rid of it, but it's kind of an interesting story, so I'll save it for that. But peroxisomes are, you guessed it again, they're vacuoles, they're sacs. It's what's inside of them that makes them a peroxisome. Anyway, all of those merge together because they're all made of the same thing, peroxisomes and lysosomes and little, what's the poem? I don't remember anyway. Vacuoles and vesicles, they're all made of the same thing. So on the right-hand side, they're merging together. Now, that has even a cooler word, autophagy, like if you were trapped in an avalanche and you got hungry and you ate your fingers. Wasn't there some story when we were kids? Oh, they buried this old guy, and then for some reason they had to dig him back up, and they realized, oh my goodness, he wasn't dead because he had chewed his fingers off trying to get out, or because he was hungry or something. Sounds like something you tell around a campfire. Okay, so that is a couple of places that one of those transport vesicles can go. So let's see. We did phagocytosis, autophagy. Any questions on that? Now, for some reason, I'm looking at my slides. I've put these out of order, and I don't know why. Well, maybe not. I'm getting ready to finish the endomembrane system, but let's talk about another membranous thing. And I've mentioned it a bunch of times already, so maybe that's why I'm thinking it's out of order. You haven't seen this slide before, I don't think so. But in the video, the contractile video one, I'll talk about that in a second. All right, vacuoles. The vacuoles are sacs. They come from the ER. They come from the Golgi apparatus. Okay, maybe that's why I put it in there. And they do a variety of things. I guess I just got my tongue kept wagging, and I actually went through this slide already. So the water vacuole, again, the central vacuole is a membranous sac, okay? Contractile vacuoles, for reasons that we'll talk about later on, a paramecium, like we saw in the picture, and those really, those cool ones we saw, in our lab, that really weird thing that I got to figure out what that was. Anyway, a paramecium, for reasons that we'll talk about later, is always dealing with water leaking into it. It's like having a leaky boat. Now, if the paramecium had no way of getting rid of the water, the paramecium, like a balloon, would eventually explode. We'll talk a lot more about how this is prevented, or not prevented. We'll talk about a lot more about why this happens later on. But contractile vacuoles have a very special function. Contractile vacuoles absorb this excess water, okay? And then they squeeze and pinch it out. These lines you see are like little rivers or tributaries that we'd be running into this big vacuole. And bloop, I'll show you a video of it in line. It's in lab. It's really cool. But that's another vacuole function, right? It's a very specialized vacuole function. As a matter of fact, I'm always getting my Bio 2 students on this. If you witness an organism like we did the other day in lab, we saw something was green, went, oh, it must be photosynthetic. If you see something with a contractile vacuole that lives in this kind of environment that I'm talking about, where you're always kind of leaking water in. So if you know anything about boats or you have a leaky one, then maybe think of the contractile vacuole as being the bilge pump, right? Okay, we're at the end now. We're ready to talk about, well, not the end so much. We're ready to talk about the last place that vesicles, these transport vesicles, might go. So does anybody have any questions for me before we get to that? So we're going to talk about the very end of the endomembrane system. I still got to talk about the recycling, but that'll be that in the next slide too. That works pretty quick. Nothing? See what I mean? Sometimes that central vacuole gets so swollen with water. All this is the rest of the cell. Those big green things are chloroplasts. Okay, so one of the other places that a transport vesicle can go would be that it's going to fuse with the plasma membrane. Now, once again, I'm hoping you're remembering, and this picture actually does a really good job of showing, all these membranes are together. So this slide, if you want to label it for your notes, is a picture now of the endomembrane system. See, all the way back from the nuclear pore, thank you, Cooper, all the way back from the nuclear pore, all those membranes are connected. They're all flown into one another. Since the cell membrane, plasma membrane, same thing. Since they're all made of the same thing, guess what you can do there? So now a transport vesicle would be like, say, a secretory vesicle. It would secrete things. Not a cool word, secretory. So, for example, in your mouth, you have an enzyme. We'll talk a lot more about this later, amylase, which starts the starch digestion. So when you eat starches, it actually starts getting chopped up in your mouth. A lot of other things doesn't really happen until it gets into your stomach. But the cells in your salivary glands, ergo the salivary cells, would be secreting, the little purple thing in that particular case would be this enzyme. You get it? So the ribosomes would make the would make the enzyme with instructions they got from the DNA in the nucleus, which delivered those instructions from the messenger RNA, which was then transported to the Golgi, where it might have been modified. Then it was pinched off into another transport vesicle, fused with the plasma membrane, and we've worked our way all the way from the code to the protein, to the, in this case, the amylase. Amylase, an enzyme, a protein, okay? So you see how that all works? Now, this doesn't mean that a transport vesicle goes to the plasma membrane. Remember, like we just talked about, lysosomes in many cases stay inside of a cell. Now, if you're wondering how this is transported the other way, if you're looking at this, and you guys are sharp, and I bet you may have some thoughts about, wouldn't that mean you would eventually run out of membrane and the cell would get bigger and bigger, right? If we're continually moving membrane to the plasma membrane, even if we take into account the lysosomes, the cell's going to get big. We know that's not good, number one. But more importantly, we run out of membrane of the ER, don't we? Well, as I said, I can explain the next part of that, the next part of the endomembrane system with this slide, but I'm not going to go into detail. But for other reasons, okay, for other reasons, other physiological events, other things that the cell's doing, there is a likewise transport of membrane in this direction too. So are things that take plasma membrane and not so much to the Golgi apparatus, but fuse them back into things like the ER and the nuclear envelope, right? So let's just kind of, that's like my professor in college talking about the Golgi apparatus. Let's just do that. Well, why? The why is because if you didn't, then you would run out of membrane, right? That's the why. Cells have to bring in things too, don't they? Like we just saw with the, yeah, like we just saw there, right? So there's a place right there. If you look at phagocytosis, that's kind of a bringing in of membrane, isn't it? So once that digestion occurs, that membrane up here might eventually fuse with other membranes or fuse with the ER. Ouch. Just punched my elbow. Cool. All right. Any questions on that? That's it for the endomembrane system. So endomembrane system net flow, or I'm sorry, not net flow is negative, but there's a flow of membranes going this way, and as we just said, there's also a flow of membrane going that way. So we don't run out of membrane, and the cells, of course, don't get bigger. Okay, here's a couple of slides that we'll move through relatively quick, but I do want to point out one thing. We're going to move them relatively quick because I'm going to talk about what they do, much more importantly in subsequent chapters. But this is the mitochondria. And for right now, let me just make it very simple for us. Mitochondria does this. Okay? Now, other parts of the cell do it, but the mitochondria is very well known for doing it. It takes the energy in food and uses it to make ATP, that energy currency that we talked about in nucleic acids, right? Much more about that later on, though. But that's all I want to say right now. So sometimes you've heard it called the powerhouse of the cell. I think the batteries. No, I've never heard it called that. But the powerhouse of the cell. Okay, so this is where that process occurs. Remember we said the other day in class, you could eat all kinds of food, and they're all, the energy in the food is all used to generate this ATP molecule. Okay, there are a couple of things I do want to point out. Number one, it's all about the membranes again. There's an inner membrane space. There's even a membrane on the inside called a cristae. The cristae refers to the, it's kind of like the reticulum. The cristae refers to the winding back and forth. And if you'll notice, let me kind of point this out to you right now. That cristae, that membrane on the inside, which is going to become really important to us in a couple of chapters, if you fold it up like the way it is, it's kind of like the bacterial chromosome, you can cram a lot of it inside of there. So think of the cristae as being not the windings as much as the membranes that are winding. So you get a lot of surface area inside of a mitochondria. Now, if you're wondering why I'm pointing that out, don't worry about that right now. Just know that it occurs because I am going to talk about it. The Matrix is that really weird movie with Keanu Reeves that I had to watch a couple times to figure out. No, the Matrix is the complete inside, but I'll talk about that later on. All right. What I do want you to get out of this here is, number one, notice that a mitochondria has DNA. That's weird. ER doesn't have DNA. None of the other organelles have DNA. Mitochondria has DNA. The only other organelle that has DNA is the nucleus, isn't it? That's kind of interesting. Let's put that down as an observation. Let's do a little science here. Because it implies then that for some reason the mitochondria needs some genetic code different than the rest of the cell, or in addition to the rest of the cell. And of course, I'm going to give you a reason why that is. But now, as I go through the slides, that what I just wrote there is going to erase. So if you want to write that in your notes as observations, if you will, at this point, then that'll be a good thing to do. Okay, shape. Look at the shape of that mitochondria. Has a bean-like Or could I say, and here's where you're going to lose that first part. How far do I got to go back? Oh, I don't want to go back that far. Prokaryotic cells. Remember bacterial cells? Didn't they have a similar shape? If you remember that prokaryote, not all prokaryotes are shaped that way, but many of them are, especially what we call rod bacteria. Y'all know what I mean when I say rod bacteria? If you just think about it, you know what I mean, because bacteria are all about shape. Well, not all about shape, but lots about shape. When you're a middle school bacteria, it's all about what shape you are. You know what I mean, Rod? Rod bacteria look like a rod under a microscope. Wouldn't those kind of be as a short, I get it, as a short rod. So couldn't you say that the mitochondria look like a short rod too? And number 3, reproduction, repro. We're going to talk about how cells reproduce in a couple of chapters. Cells reproduce. Cells need to reproduce for a variety of reasons. Number 1, you can't get any bigger. You know, as you grow from a baby to an adult, you got to have more cells. Of course, they can't get bigger for reasons that we've talked about. And also, if cells get old, they're systems. They suffer from entropy. Eventually, my buddy did have to get rid of his 72 Pinto. We couldn't just replace the door. The whole doggone car blew up, which is another story. But anyway, you sometimes have to replace an entire cell, and the cell reproduces. And when it does, all the organelles reproduce. But, this is why I wrote reproduction for number 3. As I mentioned before, if you start working out, your body, your cells, your muscle cells will start producing more mitochondria, or actually I should say it the other way around. The mitochondria in your muscle cells will start reproducing. And they'll reproduce in a fashion, in a way, it's basically called asexual reproduction, where all they do is they just divide. Simply, they divide in half. And we'll talk more about that when we get to the chapter. But those 3 observations, the fact that a mitochondria has its own DNA. Oh, no. Oh, by the way, something kind of interesting, and I wouldn't ask you about this, but by the way, the DNA that's in your mitochondria is only your mother's. It's what we call maternal DNA. In your nucleus, it's a mix, right? It's your mom and your dad. It helps us for things like tracing genetic lineage. It's not just in us; it's in other organisms as well. So, when you look at the DNA genetic information, you get a mixture. But in the maternal DNA that's in the mitochondria, you only get the mom's. Cool, huh? Okay, those 3 observations. Let's let those float in our heads for just a second, because I'm going to go on to another organelle, and those 3 observations are going to fit again. Now, this organelle is not found in all cells. It's only found in those that photosynthesize, because, again, that's what it does. The process of photosynthesis, that magical process by which, if I were talking to my kids, the sunlight energy is turned into food, is done in these organelles. So, of course, we don't have them, right? I wish we did. Maybe you could turn them on and off. Or maybe if it was just a hand. Maybe one hand. No, hand's too obvious. If you have like on your back, but nobody wants to, walk around with just your shirt off all the time. Photosynthesis would be neat. What's cool about photosynthesis, if you guys let me kind of go off on a tangent for just a second, even though we know quite a bit about it, we aren't very good at artificially doing it. In other words, kind of getting in the laboratory and artificially making a leaf, if you will, and getting it to photosynthesize. But I will ask you this question. Why do you think that'd be important? Why do you think scientists would be interested in artificially inducing or creating or doing photosynthesis? Oh, heck yeah, man. Free food. I don't know how it tastes, but it'd be free food. This is a really cool cell up above. You can see the chloroplast. This is really kind of neat because we saw them in the LED the other day. This isn't LED, but this is called, I love this name, that's why I put it up there. Spirogyra, which was a jazz group I remember years ago, but you can see why it's called Spirogyra. The chloroplasts are actually arranged in these spiral arrangements. Pyrenoids, don't worry about them. We'll talk about them. Or we won't talk about them later, actually, but you don't need to know that right now. Okay, so chloroplasts. Now, chloroplasts are interesting too, like mitochondria. They have this membrane system again. I think I may have said before a couple of times, it's all about the membranes, folks. when we get into photosynthesis and respiration, I'm going to go membranes, 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 membranes until you're insane in the membranes. So you're tired of hearing me say membranes. But you got to understand, even if you go on to A&P, or you understand, if you go on to other physiology courses, many times, like muscle contraction, membranes, membranes. Okay, so, and again, more information on the membranes of a chloroplast later on, and I'll point out what a thylakoid is, kind of a cool word. What a stroma is. You can see thylakoid are those green things, and stroma's what the space in between. Notice a couple things too. Look at that. DNA. Shape. Kind of rod-like, isn't it? And guess what? If I were a photosynthetic organism, and let's say I'm a small plant, and a big oak cover or shades me, and I get enough sun to survive, but I don't get a whole lot. But one day lightning comes, kills the oak, and the oak dies, falls over. Now I'm in what we call in ecology a light gap, right? I'm actually, maybe I'm in the forest and there's a bunch of oaks that are shading everywhere, but because the oak that died where I was, now I'm in a light gap. I'm exposed to more light. If I were exposed to more light, it's kind of like a business decision, really. If I were a photosynthetic cell, I would want lots of chloroplasts. So just like working out would make you add a lot of mitochondria, if you're a photosynthetic critter, being suddenly exposed to a lot of light or living in an environment, in a climate where there's a lot of sunlight, you want to take advantage of that, you would start making more chloroplasts. Or again, I should say more aptly, the chloroplasts would start dividing, just like the mitochondria do. The chloroplasts would start dividing. They have their own DNA. They look like a bacteria. All right, those three observations are interesting. It's not lost on just us two. Scientists looked at it years ago, and they came up with a really interesting story that I'm going to show you in the next slide. But before I go on, I want to make sure that I settle a student misinterpretation here. Both types of cells that photosynthesize, plants, and cells like ours who don't, both of us have mitochondria. Plants also have mitochondria. Plants also have a need for energy. They're a living organism. They just have the added benefit, as I've said before, of being able to make their own, right? So don't forget that. I see that sometimes on exams. I'm like, wait a minute, guys. How could a living plant stay alive then? Okay, back to those observations. I do talk a lot about this more in bio too. But this is called it's got a cool name if nothing else. I'll write it off. I'm typing it. This is called the endosymbiotic theory, endo within. Symbiosis is when organisms live in relationship with each other. Can be good. You help me, I'll help you. Could be bad. I'll eat you, you get eaten. That's a relationship. That's symbiotic. But in endosymbiotic theory, here's ingest, is it? Anyway, here's what we think happened. Sometime in the past, a eukaryotic cell, one with the nucleus, right, and the endoplasmic, engulfed a cell that underwent, it was an oxygen-using, non-photosynthetic cell. Don't worry about this part right now so much. Anyway, it didn't photosynthesize. Okay, and so then what happened is that became the mitochondria. Yeah, so this prokaryote was an organism unto itself. Remember, prokaryotes are bacteria. So what we're saying is some eukaryotic cell like yours and mine engulfed a prokaryote. Now, instead of eating it, digesting it for whatever reason, it's kind of like the elephant, the mouse, and the thorn, right? The elephant says, hey, I won't eat you or step on you or whatever the elephant does if you pull the thorn out of my paw. I know there was a story about that one time. But anyway, so they enter into a symbiotic relationship. Now, for reasons why they do this, in other words, why didn't the elephant mash on the mouse? Why didn't the eukaryotic cell just eat the prokaryotic cell? Because that does happen. That's what your immune system does, right? Big macrophages. Your immune system is full of these big eaters, macrophage to eat, right? For whatever reason, the cell decides not to do that. And now, for reasons that I will talk about later on, that cell is much more efficient, okay? That cell that now has a mitochondria has now suddenly made that cell much more efficient. If you're thinking, what does he mean efficiency? You'll have to wait till I talk about the chapter. But that's going to be actually preserved by Mother Nature. Mother Nature is going to select for that in evolution. The other one's a little bit easier to understand. What if the cell also engulfed a photosynthetic prokaryote? So now a prokaryote, a bacteria, but now it can photosynthesize. And what I mean by this one's a little more obvious, the advantages, right? So now that photosynthetic eukaryotic cell can photosynthesize. Don't got to go find food, right? So if you think about it in very broad terms, leading from here would be the animals, the fungus, oh gosh, I open up a can of worms. Some of the protozoans, and this would be the plants. There are some algae that can photosynthesize. All right, see what I mean? So this endosymbiotic theory is really kind of cool to me anyway, because it's great about science. It's an observation, based on what we saw. It's hypotheses. Now, can we prove anything like this? We have a lot of circumstantial evidence to support the evidence, but come on, to support the theory, but you're not going to find something like this happening in the fossil record. Soft things like cells don't fossilize very well. Usually it's bones, okay? Now, if you're really fascinated by that, I can talk more about it with you over email or in class, but also in my bio too. Yeah, this would be an example of evolution. Evolution supports cells becoming more and more efficient. And when that cell engulfs the mitochondria, or I'm sorry, that thing that becomes the mitochondria, or that thing that becomes the chloroplast, then it becomes more efficient. There are poisons in your body that your body makes, right? Not just poisons you introduce into your body, but there are poisons in your body that are a necessary part of life. You can't do anything about them. This is something called a peroxisome. Peroxisome. There you go. And a peroxisome, when you see the root some, like chromosome, it kind of roughly refers to something to do with a cell. So this does. It's a cell organelle. It's a part of a cell. The first part of the word, Mike. Right? I didn't mean to put the slash. Peroxide, right? Hydrogen peroxide, H2O2. It's kind of a weird type of water. H2O2. Hydrogen peroxide is really good for treating cuts. Not real good for treating punctures. It's really good as an antibiotic because it's a heavy oxidizer. And we know what heavy oxidizers do, right? Oxidizers tears things apart. And if you have some bacteria living in a wound, you dump some oxidizer on there and you oxidize it. The stuff you buy in the stores is, I think it's 3%. The stuff we used at SeaWorld that would actually dissolve the meat off the bones of the whales after they died was 30%. Dissolving the meat off the bones or killing bacteria is just oxidizing, all right? Now, you'll notice that if you look right next door, there's a chloroplast. This is a chloroplast. Okay. So what's happening inside of this cell is hydrogen peroxide is accumulating. I'm sorry, not inside the cell, inside the cell organelle. And again, I'm sorry for the blue and green. But a dangerous substance, because an oxidizer like hydrogen peroxide will oxidize, anything I've talked about so far, including the cell that it's found inside. But notice, of course, that luckily it has a membrane around it. And if you're thinking, well, don't membranes merge? Well, usually they do, but sometimes they're reinforced for such a way that they don't merge. And that's a good thing because inside this organelle, hydrogen peroxide builds up. And if hydrogen peroxide were allowed to leak out of those membranes, it would destroy things like mitochondria, it would destroy chloroplasts, it would just wreak havoc on the rest of the cell. It is separated by a membrane, yes, made of the same thing, but for reasons beyond the scope of the course, that membrane is set up in such a way so it's not permeable. It doesn't allow that hydrogen peroxide to leak through. Now, if you're thinking, where does the peroxide come from? When you metabolize fats. When you use fats for energy, one of the byproducts is hydrogen peroxide. It's chemistry, folks. There's nothing you can do about it. Chemistry, laws of chemistry are in place before living things, right? So if you want to metabolize fats, because of the chemistry needed or involved to do that, you're going to produce hydrogen peroxide. Luckily, and we'll actually do this in lab, there's an enzyme in peroxisomes known as catalase. And what catalase does is it breaks down hydrogen peroxide. It turns into water and oxygen, which aren't damaging in their own right. Oxygen can, then be used by the cell actually. So even though oxygen is obviously an oxidizer, hydrogen peroxide is actually worse of an oxidizer, if you can believe that. Okay, so this is the important thing about talking about peroxisomes, more than just, to me anyway, more than just the idea that they help detoxify, is the fact that membranes can be variable depending on their needs, right? That sometimes they blend together, and sometimes for reasons of the way they're structured, they don't blend together. And membranes are entrance, right? Either you go in the cell or out of the cell. We talked about that in nuclear membranes, didn't we? Any questions on that? I'm now going to talk about a collective of organelles, I guess. This is the picture I had at the beginning, which if you started the recording, you don't see because I forgot to turn the recorder on at that point. Recorder on? Yes. This is actually a cell. The blue thing in the middle is the nucleus. And all of the spindly things you see shooting out everywhere, they've been colored by a computer, obviously. Those are, which all look like a fibrous network, don't they? You got ones that kind of go around the outside, the orange ones, and then you got these green ones. It looks like, if you will, if you let me, a scaffolding maybe, a structure by which to build a cell on. I'm not too far-fetched on that, right? And there's a reason why it looks like that. Its form, if you will. Its function is the cytoskeleton, right? These are, what you see here is a color-coded picture of a microscope image of the cytoskeleton of the cell. What they do is they get each of the parts, they give them a radioisotope. They impart a radioisotope into their chemical structure, which glows a different color. Isn't that cool? So they make the nucleus glow blue, and they make the parts of the cytoskeleton glow green, or in this case, orange. Okay, let's look at the different parts of the cytoskeleton. Let's look at the orange parts and the green parts, and a part that's not labeled on here, actually. Things called microtubules, okay, microtubules. And you know what? Don't worry about this. Helps if I turn the pen on. Don't worry about this. Don't worry about this. Has to do with their chemistry. If you're interested, a tubulin is the name of a protein. A dimer means there's two of them. There's an alpha and a beta, and they stick together to make a dimer. And then you wrap those things up and you make a tube. Microtubules, tubulin polymers, right? Polymers of tubulin. They're tubes, okay? What do they do? Well, one of the things they do is they act as the girders for a cell. So basically, kind of like our skeleton, they help hold up the cell, if you will, right? Cells don't have a skeleton made out of bone like ours, but anything rigid, even at this relative level of a cell, can hold them up, right? A little bit later on, we'll talk about microtubules also make up these things called cilia or flagella, which was really cool because we saw them in the microscope the other day. Definitely saw flagella. That's flagella and cilia are the components, the little wiggly things that may look like tails or hair, if you remember from your earlier bio classes, that move cells around. Cell motility, movement. Later on, when we talk about cell division, these are the chromosomes down here in this picture. Isn't that a cool picture? Again, blue on blue, sorry. But microtubules are the green things. They're responsible for moving chromosomes around. Okay? And I'll talk more about that later on. I don't want to get into the detail of the function of that so much, just the form. Organelles also need to move around inside of a cell, and the microtubules are responsible for that. Many times we talked about how things randomly just float throughout the cell, and they do. But in some cases, there's a reason to move something from point A to point B. This is so cool. Gosh, you guys got to see this. Think about these microtubules as being monorail tracks. Okay, monorail tracks like out at Disney. Here's how it happens. So let's imagine inside that vesicle up at top, we have to move something. And we have these proteins, that's what's in purple, those little purple balls, that are receptors. So think about those as being a connector, yeah? And that receptor connects to a thing called a motor protein. You see there? And that motor protein is attached to that, the Disney thing. Anyway, it's attracted to that tubule like it's a track, right? It's a tubule, a part of the cytoskeleton, a microtubule. Now that motor protein, guess what? Is powered by ATP, which is energy. So what you can get it to do is you can get it to move. See, a motor protein has a shape. Now, it may not be shaped like two little legs like you see there, but you notice it down below. It doesn't quite look that way in the real picture. Isn't that a cool picture? That's a giant squid, or a squid giant axon. An axon is a nerve cell. But that energy causes that motor protein to change shape. It's the most important thing about a protein. And guess what that thing does? It causes it to change it so that it walks. You see the little pictures how it's the jagged part? It's showing you that it's walking, it's moving. So it's moving that vesicle along. Isn't that cool? Isn't that neat? Little vesicle. So if you notice when you were looking at the Elodea yesterday, you might have saw something called cytoplasmic streaming. You might have saw those cytoplasm moving around, mostly on the outside of the cell in kind of a circle. If you're interested in that, I think there's a video on Blackboard. If not, you can easily find one on YouTube. Those little chloroplasts that are moving around in that cytoplasmic streaming, that's how they're doing it. They're walking. They're being moved along by these little motor proteins. So cool to me to think there's little things walk along with these little vesicles, right? Isn't that neat? Okay, so those are microtubules. Those are the tubes. Those are big things. They do lots of things. They help move things around, like we see here. They pull chromosomes apart. They're a little bigger, a little thicker, a little heavier, a little more durable. So they act as girders, kind of hold the cell up. Okay. All right. Now the next thing we're going to talk about are the microfilaments, or something we call actin filaments. These were the things in the picture. I think they were colored orange in the picture. Or no, I'm sorry. No, were they? Yeah, they were the orange things on the outside of that picture at the beginning. Right there. The orange things there, the red things here. Okay, they're not microtubules, are the green things, remember. And so, here we go. Microfilaments, actin filaments. Actin filaments, because that's the protein they're made up of. They're made up of a protein called actin. Again, don't worry so much about size. Actin is the protein. You see the way it is. It's kind of like it's twisted together. Now, actin, like lots of proteins, is stretchy. So think of these actin filaments, these microfilaments. Think of them as being rubber bands. They're stretchy. All right. So what does this do? Maintains the cell shape again. But this time it's not like a girder, right? It's the part of the building that's flexible, right? Oh, gosh. If you lived out in California, the buildings are built to withstand earthquakes. And the way they do this, they make them flexible, if you will. Right? A cell wants to maintain a shape, but it doesn't want to be rigid. Especially like, remember we talked about how red blood cells, how they pinch through capillaries smaller than them? It's the actin filaments that let them do that. That's why the actin filaments are on the outside of the cells. They surround the perimeter. What makes the cell stretchy, allows it to change shape. Muscle contraction. If you get on into A&P, you'll learn in A&P that there they are, actin filaments. Actin filaments help your muscles contract. Now, I'm not going to go into muscle contraction. You'll learn about it in A&P, but it's very, very cool the way it works. Muscles only contract. When you take your arm and you bend it, your bicep contracts. That's why your arm bends. And then you use your tricep on the other side. You can't push with the muscle. You can only pull. So what these myosin filaments do, they have little heads on them. See little bulbs right there? And they act like a ratchet, and they pull those actin fibers close together. So when muscles contract, those actin fibers overlap. That's how they get shorter. The cytoplasmic streaming, I mentioned to you, those transport vesicles on the outside, those actin filaments also help with those microfilament, or with those chloroplasts as they move around the plant cells. So it's not only those tubes with those motor proteins, it's also these little actin filaments that help bounce that stuff around. Amoebas, oh gosh. Amoebas are really cool, but they're so hard to help folks find under a microscope if we're using pond water, because it's like a blob. Amoebas don't have any definite shape. They kind of move around as a blob. If you look, you might recognize that as being That might be the nucleus, or that might be a contractile vacuole, because they also live in areas where they fill up with water. But the way an amoeba moves is through something called pseudopodium, false feet. Pseudopod is foot. We're all familiar in Florida, especially in the summertime, with these guys, arthropods, insects. Insects have jointed arthroscopic knee surgery. Insects have jointed appendages. Anyway, amoeba is obviously not an arthropod. It's a protozoan, but the way it moves, if it wants to move, say, in the direction the arrows are pointing, it takes this inner cytoplasm that's much more fluid, and it pushes up against this outer cytoplasm. It's like a gel. In the result, the membrane begins to stretch out, and then the rest of the amoeba follows behind. The fact that membrane can stretch out, right, without breaking. Think about a balloon, folks. The fact that you can blow up a balloon without it exploding is due to the elastic nature of the rubber in the balloon. The fact that the amoeba can move along that way is because those actin filaments are stretchy. Now, amoeba could eventually stretch to the point, but it wouldn't do that. It wouldn't be very smart. Not that they're smart anyway. I'll show you some pictures of amoebas moving around in lab too. Okay, the intermediate filaments. Where are we in time? Oh, good. Write myself a note again. I got to reschedule exam 2. All right, intermediate filaments. Again, intermediate filaments are made of proteins called keratin. If you're going, I know what that is. Where do we find keratin? Where do we find keratin proteins? Or keratin? Yep. Fingernails, that kind of stuff. Yeah, there you go. Again, don't worry so much about the size, but these are cables, all right? So as you can see down below, they're keratin proteins kind of wound up in what we might call a helix, right? A helix being like a spring. This is, again, kind of like the actin filaments, not so much the girder thing, but allows a stretchiness. You can almost imagine looking at those keratin proteins, keeping in mind that proteins can be flexible, might absorb the shock of the cell being pushed around. Certain organelles, like the nucleus, for example, they don't really move around the cell much. For one thing, you generally want to keep that in one place. So these intermediate filaments can kind of act like, if you think this is the nucleus, they kind of act like oh gosh, I don't know a good analogy, but a framework that holds certain organelles in place. Oh, this is a cool picture. I remember this now. Why I put this in here. If you take a look picture down below, this would be an electron microscope picture of the cells that line your intestines. There's a couple of really cool things we can look at here. Cell membrane, you know what that is. Microfilaments, just talked about that. Look, intermediate filaments. Intermediate filaments actually can provide, since they're kind of strong and stretchy, they can provide a foundation. So the nuclear lamina, remember we looked at the nucleus and it looked like it was fibrous? That was made of these intermediate filaments. Now, in the microvillus that you see there, the microvillus are the little fingers. In your intestines, the cells that do the absorbing of food from your intestines, if you look at the part that faces into the lumen of your intestines, the opening, under a microscope, oh, come on. I always forget I have to do that. It winds back and forth. So that would be a cell. And of course, as you see, the intermediate filaments would anchor it down. But my question to you is, what's the purpose of intestinal cells, as this bagel that you ate for breakfast floats by, what is the purpose of having the windy back and forth, the microvillus, the microvilli, I think is plural of that. What's the purpose of all those little fingers? If you think about form and function, it does help do that. I'm sorry. Yep, I should have qualified. Yes, it does help move the food along. It kind of flows. But as far as the purpose of the intestines, what do those little wiggly things, or what do those finger things do? What do they increase? Yeah, sure they do, Tim. Right. Take the same size cell and compare it to a cell like that. along this surface right here. There's that bagel again, right? Which one has more surface area? Obviously, this one does. Form and function. You're looking at a bunch of cells with a bunch of microvilli. Ooh, I don't know what they do, but I'll bet you it's absorption. See? It's not magic. Despite what I told my kids. All right, so we're still sticking with intermediate filaments, but I'm sorry, we're still sticking with the cytoskeleton, but we're going to look at some other ways that the cytoskeleton, or maybe I should say some specific ways the cytoskeleton is used. Now, in red, I put this in red for a reason, because I want you to remember, in animal cells, and this only exists in animal cells, plant cells do not have them, there is an arrangement of microtubules in triplets. You can see there in the picture. It's like 3 attached to a group of 3, attached to a group of 3. In animal cells, those occur in pairs. A pair of them, with the dashed lines around, is known as a pair of centrioles. You can see them in the microscope down below. They always come arranged where one is up and down, and one is, I don't know, left to right. One is laying down, one is standing up, depending on how you look at it. Now, what do centrioles do? Well, we'll talk about that when we get into cell division. They seem to have something to do with cell division. I seem to because, I don't know, maybe the jury's out on that a little bit. I'm not sure if the current literature suggests that they finalize what these things do, but we can certainly suppose what they do. What is a centrosome? Okay, a centrosome is kind of like a shell. There's no such thing called a centrosome. It's the area where those centrioles are found. All right, so animal cells have centrioles in pairs. Now, plant cells have microtubules. Don't confuse that. We're saying that only animal cells have them arranged in this pair of centrioles in this centrosome region. Purpose, TBD. Talk about that later. Okay. We can talk about the purpose here. In certain cells, animal cells, but many protozoan cells too, who have cilia and flagella. All right, let's start off very broad. When an organism has cilia are usually shorter, thinner, and cells usually have lots of them. Cilia are usually covering the cell. If a cell has a flagella, or flagellum, I think flagellum is singular, flagella is plural. I always get those mixed up. You either have one or a few, maybe sometimes a half dozen. You don't have very many, and they generally tend to be longer and thicker. You can sometimes see cilia under a microscope. You can sometimes see flagella. Flagella are a little bit easier depending on the critter. Although I have some great videos I'll show you in lab where we can see some cilia and flagella. Now, cilia and flagella, as we said, are for moving cells around. That's their job. Either little tiny cilia or big flagella, when they wag back and forth, they propel cells to their liquid. When you looked at them under the microscope yesterday, that's what they were using. If we looked at a cilia or a cilium, that's a singular, we would find something very interesting. A cilia is made up of microtubules in that triplet, just like we see in the centrioles in the animal cells, right? Same sort of idea, different kind of function. But we see the same sort of arrangement. Mother Nature is the classic recycler. If something works in one place, if you can make a structure out of it, then, don't reinvent the wheel. Now, there is a difference, though. Really cool difference. Remember those motor proteins? Those things that can walk along the microtubules? There they are. This is how cilia or flagella bend, folks. Down below, I'm showing you that that mechanical device will only allow that wheel to go in one direction. So when that cilia, if you imagine this one, wants to bend this way, those microtubules on that side, those motor proteins, they will shorten the distance between those microtubules. So they'll walk down or up those microtubules to get that cilia, which I could show you with my hands. But if you took your hands and you left your wrists limp, you could push back and forth on your arms. You can see you could wag something back and forth, couldn't you? So what you could do, if you took something that's here we are, and you shorten and lengthen it on opposite sides, oh my gosh, I am a horrible artist. It would wag back and forth. I'm trying to think of a good example, but I think you guys understand what I mean, right? That's how those motor proteins work. They shorten and lengthen those different sides of the and the fact that they do it so fast, that's what gets to me, right? So we can find things like cilia and flagella. Like I said, you're going to find flagella typically one. Sperm cells have flagella. Sometimes they have more than one, and other organisms have lots of cilia. Down below, that's a paramecium, what it would look like under an electron microscope. And you can see that even though you couldn't really, see it under the microscope yesterday, they're like covered in these little cilia. All very coordinated. It's very cool how they work. Here's an interesting picture. This is an egg cell, not human because those aren't human sperm, but all those sperm are trying to make their way into the egg cell. There's all kinds of interesting stories about that. And I thought this was an interesting little quip down below. If you scaled up sperm, they'd be dangerous as salmon, wouldn't they? And they'd definitely be dangerous if they were whales. So sperm are definitely in a hurry, aren't they? So in their own little world, they're zipping along at 500 miles an hour. Of course, they're not moving at 500 miles an hour. You're scaling here, but that doesn't mean they're not in a hurry. They don't last too long, and everybody wants to be the first one to get to the egg. If you want to know more about sperm competition, the fact that there are oh man, I wish I had time. I'll talk about it if you remind me in class. I am looking for India ink. I couldn't find it the other day, but I'm going to see if I can buy some. Oh, we only got 1 minute. Yeah, no, I'm going to stop here. Wait, I do have 1 minute. Let's talk about sperm competition. Interestingly enough, when males ejaculate, that deposit of sperm has different types of sperm. The first type of sperm that come out of the ejaculation actually give their lives up, if you will. The vagina is a relatively acidic place. It's acidic because that's good for keeping out infections, right? It makes it not very hospitable for bacteria. Well, those first sperm that come out are actually responsible for neutralizing the acidity, buffering it, if you remember that word. The sperm that come behind, what they do is their job is, of course, to seek the egg. And you've got two fallopian tubes. The egg's only in one, so if you go up the wrong one, you're out of the loop. Now, what's really cool is there are Rambo sperm. Rambo sperm, you don't know who Rambo is, you can ask somebody older. But anyway, Rambo sperm hang behind in the vagina, and their job is to destroy sperm that enter in after them. So what they try to do is prevent another male's sperm from doing the fertilizing, right? So in some cases among female mammals, if they mate quick enough in succession with different males, this is a way to help prevent the male that comes after me from its sperm making its way to the eggs. Because, among most mammals, not so much in humans, but among most mammals, the many eggs are deposited during ovulation. We generally only ovulate one. Isn't that neat? Rambo sperm. Very cool. Okay, with that message, with that thought, I'll leave you for the weekend. As I said, I will be working on your exams. What lab are we doing next week? We're doing 5, right? That should be online, so make sure you download it. I am going to move the exams, more on that later, and I'm probably going to move the lab practical too, but I'll think about that, and I'll send an announcement out, okay? If you have any questions, on sperm or anything like that, you're more than welcome to stick around. If not, please enjoy your weekend.